Welcome, everyone. Thank you for being on this panel. I think we have 3 speakers here. Dr. Jiang, no need another round of introduction. Associate VP for catalog product at GenScript China. Tsutomu-san from Takeda Global Engineering, as well as Zhao from Dark Horse Consulting, General Manager for the Asia Pacific region. In addition, we welcome back Dr. Zhao, right? Zhao Yuan from Hillgene, which is actually a CGT-focused CDMO in Jiangsu, China. You are also a speaker this morning talking about CMC for cell and gene therapy. Thank you for that. And next to Dr. Zhao, we have Dr. Kitipong from Genepeutic, which is also a CDMO focusing on CAR-T, and maybe there will be interesting expansion plans. I'm just guessing in the future. Hope to hear the good news from you from Thailand. Last but not least, right, our very own, a friend of ISPE. Not sure if you join our society. Hopefully so. Dr. Sissi Lin. Sissi is the director for R&D strategy at NATi. So just a little bit of a commercial time for NATi, standing for Nucleic Acid Therapeutics Initiative. So this is one of the national platform funded by the Singapore government to focus the development effort on nucleic acid therapeutics. So as you can deduce from the label, right, including oligonucleotides and mRNA side of things. So welcome everybody to this panel. So we are going to kick off with a little bit of a landscape, right? So what it really means to Asia Pacific, to our patients, and how can we expedite the wide accessibility of the life-changing drugs, right? So I'm just going to read out some numbers that I got from American Society for Gene and Cell Therapy. By the way, just go and download that. They have this landscape reports published every quarter. So from quarter 2, they're saying now globally we have 31 gene therapies, 30 RNA therapeutics, mRNA oligo included, 68 non-gene modified cell therapies. So the number is quite impressive. And John, you're also showing, actually presented the uptake, right, in terms of approvals year on year. It looks very promising. And I think being in Asia Pacific, we should be proud, because guess what? The first 3 ever approved cell and gene therapies happened in this region. So 2 from China, 1 from Philippines, right? Those are oncolytic, if I remember correctly. However, in recent years, especially this year, we heard about 2 T-cell therapies for solid tumor. So one is actually the tumor-infiltrating lymphocytes, TIL, from Iovance, and the other one is actually from Adaptimmune. So it's the first TCR-T for solid tumor. So there's a sentiment that we still need to catch up, right? We want to be, in many cases, I think the mentality is a fast follower, right? So we want to do me too, hopefully not me worse. But I really want to, just start this panel from John, right? So, on the consultancy side, you have seen a lot of things, right? So I just want to have a provocative question to you. What is really the unmet need? What does it mean in terms of developing ATMPs for the the Asia Pacific patients?
Yeah, I think firstly, drug development is a very expensive endeavor. Really, a lot of drug development goes hand in hand with the VC dollars. And because of that, you start seeing all the first-in-class developments happen in the US, where they have the payer system, where they have the ability for an approval by FDA, and then all you have to do then is to negotiate with the PBMs to actually set the price of the drug. And without that, it's very difficult to try and develop a first-in-class drug in APAC. I think that we have done well is that there is good science coming out from APAC itself. The challenge is that with a first-in-class, there's high risk, and the question is whether the investors will want to invest in this moving forward. And I think that's something we have to think about. So if I look at it, I think it's going to be a 2-stage process. Long term, we focus still on first-in-class, but I think that there's something to be said about best-in-class, right? In the short and midterm, maybe that's the one thing that we have to work on. We should think what are the advantages we have here in APAC, which is a very competitive cost structure. We have lots of patients in APAC. These could be some of the advantages that we have to first, as a first step, try to reduce the cost of these advanced therapies, which are, you know, $500,000, and try to make it more accessible to everyone here. Once you're able to do this, you're able to actually drive adoption, and I think that will then set us up for moving from the best in class to the first in class. Yeah.
Thanks, John. That's a very nice, kind of a high-level roadmap, right, leading to the Asian story for ATMPs. Now, turning to Sissi, right? So now you are, directing the R&D strategy for NATi for Singapore. I really wanted to hear from you. What does it mean, right? What is the quote and unquote unmet need, or loosely speaking, what is your business case? What do you want to develop for Singapore?
Thank you. I think John has just mentioned that the drug development mostly is done in US and Europe. So the data, particularly the patient data, are mostly collected among the populations there. So I think for Asian, actually there is a strong unmet need because there is a variation from the clinical symptoms and also the genetic variations. So I think one word that we have been discussing a lot in Singapore context is Asian biology. What is the real Asian biology that we are having here, right? So I think there have been lots of research demonstrating that for metabolic disease, and there has been lots of differences among different races. For example, like diabetes. Diabetes in US and Europe is mainly driven by obesity, but in Asian countries, you can see it's mostly can be very genetic driven. And also for oncology, the variations in Caucasian population and the Asian populations are very different. So how can we address these issues by having our own translation, basic science, and also translation research, and be able to enable the drug development in this region, right? So I actually want to mention one initiative on the basic science part, because data is actually very key. Understanding the disease, understanding the patient is very important. So that's why in 2022, Singapore government started this precise SG100K to sequence 100,000 Singaporeans' gene data to be able to find out if there is any specific targets that can be very different from the existing data. And these basic research actually can be the driving force for the drug development in this region. Yeah.
Yeah, that's a very nice perspective. So just hang on for a while, Sissi. Just another follow-up questions for you, right? So you mentioned about the newborn screening, the kind of genetic screening, right? All those data that will become available. So it actually reminds me of a program at the international level. It's called NLORM. I'm not sure whether you heard about it. So this is really targeting the nano-rare diseases, N of one, right? So like one patient or, very small patient population, right? So the idea is there's a consortium being formed, manufacturing capacity, suppliers, and also the hospitals, including the genetic sequencing capabilities altogether. Once we identify that newborn, ultra-rare, nano-rare defects, then we could hopefully apply a sort of antisense oligo, so that's in your area, right, to treat those kind of diseases. So whereby, in this consortium, actually my organization, Cytiva, is actually playing a part as well, providing technologies, consumables to enable the program. So I just wonder, now you mentioned that, are you alluding to, some sort of a Singapore version of an NLORM program, or what do you see about the opportunity in that space?
Okay, I think just now I mentioned about precise 100K, which is more about the basic science discovery side. If it's talking about the drug development side, I think we can look at the two major national platforms in Singapore. One is EDDC. The other one is actually the newly launched NATi, Nucleic Acid Therapeutic Initiatives. So for us, what we are really looking at is to look at the target discovery that has already been quite mature in Singapore, and potentially to leverage on build up the capabilities to set up the modality and molecule translation capabilities in Singapore, so that we can potentially translate the targets into molecules and address the unmet clinical needs that we just mentioned. Right, so the modalities we are looking at right here would be mRNA, antisense oligos, and the siRNAs, these relatively more mature modalities. But we will also have a certain focus on the novel modalities, potentially can become the next generation, but we will mainly rely on the community, the research community, to develop these further.
Thank you. So now I'm going to pivot a little bit from nucleic acid therapeutic side of things, whereby I know we can realize like kilogram level production for oligo and very high productivity for mRNA, potentially drive down the cost of goods tremendously. But over to the cell and gene side, especially CAR-T side of things, right? We know the manufacturing cost of goods still very high. The manufacturing process is still cumbersome, right, and highly variable. Variability, that's the keyword I saw in everybody's presentation. So maybe let's delve to that point a little bit. A question to first, Dr. Jiang, right? So you led the CMC development for Carvykti all the way to marketing authorization. So over this development path, if I just pressure cook a little bit, right? So name top 2 technical hurdles that you've encountered, and what is your workaround, or how do you totally resolve it, hopefully?
So technical hurdle for the CAR-T, like we develop, is like how to we set the spec of the in-process control and the final batch release. Because originally, so when we do the test on the patient in China through IIT, we did 72%, or 72 patients. So 72 patients, like in different hospitals, and also the dosage is totally different. So how to decide the dosage at the beginning of this product, and also the spec, because actually the IT is in the IND stage, so we do not have a very strict like the criteria at the beginning. So I think this is the biggest 2 hurdle like we are facing during the Carvykti development.
Thank you. So my takeaway is spec setting and dosing, right?
Right,
Initial
yeah.
dose. Okay, that's a fair point. Yeah. So over to you, Dr. Kitipong. So Genepeutic, right? So you guys have started up, and congratulations for getting the GMP certification and using our end-to-end technology. Not forget to mention. So as a kind of a solution provider, being a CDMO, right? So you must seen different products or processes from different donor sites, right? So to you, what does it mean, right? So again, name 2 most significant hurdles to you.
Well, I guess as a GMP certified facility, right? I guess the first hurdle will be obviously starting up a GMP facility first, because going through with the regulator, make sure all your processes are validated accurately and correctly according to the regulation there. It's quite a big hurdle really, because as you know, cell and gene therapy is pretty new, especially in country like Thailand, right? Where the regulation is not really set in stone compared to like countries like in the US or the Europe, where they already have the experience of approving quite several products in this field. So I think that hurdle that we have to actually jump through, it sort of like taught us quite a few lessons on, maybe we should actually start up like learned together with the Thai FDA and come up with a regulation, etc. So that's one of the hurdle. And the second hurdle will be obviously what technology should you use really? Which one will be more compatible to first of all the regulation in Thailand, where they I know 3-4 years ago, they really regard GMP facility as being one room, one function. So obviously that will eliminate the fact that you will have to, bring in a system where it will be all in one system, right? So that will tend to facilitate the approval much quicker compared to, trying to force something that they don't believe in. But I mean, as the trend now is moving towards a different strategy, like a lot of companies is actually setting up like a all-in-one machine that will obviously change things a bit. But I think that's those two are the hurdle that we actually are facing. Yep.
Yeah. Thank you, thank you. So talking about regulations, that's a very nice segue to you, Dr. Zhao, right? So you have worked, spending years with the UK agency, MHRA. You have been, subject matter experts for BP, USP, and a series of, advisory committees and, international bodies. So, and now you are in China, leading this Hillgene platform. So based on your observation, could you name a few key differences in CGTP, cell and gene therapy products regulations in Europe or UK included versus China or other regions that you are familiar with?
I think the regulation is maybe just in Europe is more matured because the guideline development since 2004, I think, is already starting. Like in FDA is starting from 1998. So and the whole theme of the regulation, I think, is similar, like between China or also it's now is all sort of like following the development and the requirement of the whole field. And from the earlier questions about the what I was thinking about what's need for the Asian Pacific area, I think there are two aspects from my personal view. First of all, is I think maybe for the whole world, is reduce the cost. Because everyone recognizes that this therapy is very promising, it's really worked. Yeah, so that's why I believe that every government, even the European, even different country, they invest a lot in encouraging a lot in this area. So I think the team up to reduce the cost is a must for the whole field. And I think it's very probable because before the cost high is because the lack of experience in the manufacturing and testing, so it's incurred like a lot failure, the batch failure. So that also added to the cost. And I think there's some of the two manufacturing process, two products at the beginning, maybe 50% of the successful rate. So that means half of the product is failed because the patient variation, the manufacturing immature, manufacturing process. So I think, and also maybe now we're moving to the automation and also the equipment. I think maybe early on is more manipulate. Maybe some company like have certain equipment, so the price is high. And now I think the equipment side, a lot of company get involved, so that will reduce the cost. So I think that the first thing is to reduce cost. The other thing is what I'm thinking is since the government is really encouraging this field, I feel like everywhere, like even in the Asian Pacific area, right? So I think maybe you should lobby the government, have a new kind of the payment regime, schedule or strategy. Because I think because we don't forget, gene and cell therapy, there's a one-off treatment. They're supposed to be only receive treatment for once and then have the permanent cure. That's the aim. And in theory, it should work, right? Particularly for gene therapy product. So I think that's why the cost is to spread the whole lifetime. Such as for the factor IX, the product. If the patient doesn't have the gene therapy product, they might pay for a huge amount, maybe more than the gene therapy product. And then the patient suffers as well because they need to continuously infuse the factor IX, the product. So I think maybe one is the reduce the cost. The other one is the new kind of payment strategy. Maybe that will speed up this kind of therapy and then also meet the patient's need. Yeah.
Thank you, thank you. So, in my opinion, the cost is actually out of regulatory consideration. However, I just recall, the FDA CBER director, Peter Marks, he was basically calling for pulling all levers, that's the word that they use, to streamline and accelerate ATMPs, right, cell and gene therapies. So if we wanted to take, in part its therapeutic functions, we need to achieve that, streamlining and, acceleration in 20 years, right? So of course, the ripple effect will be, cost reduction, et cetera, et cetera. So talking about manufacturing cost of goods, question to Tomu-san, right? So, and also in connection to what Dr. Zhao shared this morning, where I asked you a question about the point of care distributed manufacturing. So again, on the provocative side, Tomu-san, do you believe for autologous CAR-T, point of care manufacturing is the future?
So I think yeah, I think CAR-T manufacturing, currently centralized manufacturing, they are very common in Japan. But I think for the future, it's also autologous. Sometimes people want to have a kind of distributed manufacturing process, but it really depends on. So I think always, investment for CAR-T and the autologous area is very hard, and especially ATMP area is very hard. Yeah, so we always, have to think about kind of process, capacity demand, future demand, and also, so what kind of modality? There are so many approaches. There is the scale-up approach and the scale-out approach, and the autologous, and also allogeneic approach. So I think an important thing is I think facility how facility has a kind of expandability and flexibility for the future demand in the future. So.
Right. Yeah, that's a good point. May I also ask you, since you are chairing the ATMP COP for ISPE Japan, is cost modeling or cost calculation estimation, is this part of your work stream or work package? Is that something you guys are working on? Short answer, yes. Okay. So yeah, that's a good one because really, I feel sometimes it's not straightforward, right? Cost of goods, is typically a confidential information, right? So, but really encourage within the boundary of what we can share, right? So we can heavily redact it or we can generalize it, but the form of sharing is really important for us to collectively move the needle for this industry. So question to Sissi, right? So I think yesterday we talked about how to sustain the pharmaceutical manufacturing. I heard my personal takeaway, 2 keywords, right? So I remember one of the panelists, so he talked about a common stockpile, and then there's a apparently contradicting keyword, dual sourcing, right? And I just want to enlarge this problem statement a little bit. In the current, macro environment, what's happening in the world, we tend to see a lot of sovereign biotech, sovereign manufacturing, right? And apparently you guys are doing something where, when John shows the global footprint of CDMOs, apparently there seems to be a number of CDMOs available, nucleic acid therapeutics included. So perhaps a question in terms of differentiation or, overall organizational objective, right? So what do you guys do that is different from, a true blue CDMOs or CROs, and what do you really want to achieve at this national platform level?
Sure. Yeah, I think the whole idea of nucleic acid therapeutic initiative was really to build the resilience toward the pandemic. So because through the pandemic, we have seen that in such isolated pandemic world, everyone has to protect their own supply chain to enable for national security reasons. So we had basically in Singapore, we had the similar idea. That's why the initiatives was started to look at an end-to-end process, including the design, all the way to the manufacturing, right? So these capabilities are very new to Singapore. So that's why we are putting lots of investment into this and building these capabilities up. But actually, along the way, we also realize By enabling these capabilities, it will not only just, of course, it will build up our resilience to the pandemic, but at the same time, there is a huge opportunity right here that we can potentially translate a very strong research community that we have in Singapore and all the Asian biology, APAC special strength that we have, right, and translate it into economic value. That's also one of the reasons that we further expand the nucleic acid modalities from mRNA to include oligonucleotides. Another point right here is personalized medicine is also something that could be very interesting for particularly for cancer patients. I just had quite a few conversations with cancer doctors recently. I think they have mentioned that for mRNA treatment or even like cell gene therapy treatment, right, they are looking at 6 to 8 weeks window. So how can we, basically they are going to pass the researchers are going to get the patient samples. Starting from that moment, we have the clock is counting, right? So how can we make sure that we design and also including all the development stage and the manufacturing stage, assure everything is under well-controlled, like even GMP standards, right? How can we ensure the whole process is well done and then be able to deliver the therapeutics to the patient and improve the care, right? So by thinking about that, I think the end-to-end process has a great value in supporting all the 3 different perspectives. Yeah.
Yeah. So we would really like to, have you back next year to give us an update on this very interesting initiative. I think that's really one direction to go. Clearly, the need is there, right? So now let's change gear a little bit. We talked about, we heard, from multiple presentations about changes, variabilities, clinical development, and the resulted comparability, right? So the need to demonstrate comparability and your strategy, right? So I would like to pass these questions around this panel, maybe starting from Dr. Jiang, right? So, knowing that there are several comparability strategy guidelines for CAR-Ts, for, ABC, different types of ATMPs, what does it mean? Give us some practical guidance, right? What does it mean to CMC non-clinical and clinical?
So for the change for the CGT product is inevitable because facility change, right? So, and also possibly you want to constantly process change. And the issue for the CGT products, when you develop it, so usually for the critical key material, you have backup. But a lot of the CGT product even don't have backup. You have the primary one. So possibly in the later stage or they have the backup showing up, right? So the compatibility change, so change is inevitable. So when do the change? So you should understand your product and your process well. Because if you understand your product process well, it will help you to evaluate the post-change, how affect the post-change to affect the safety and quality of a product. So you should do the risk assessment and also decide if it's the major change, moderate change or minor change. So for the CAR-T products, so I compare to the comparable study. So for me, usually if the reagent change, right? So lentivirus change is possibly it's huge change, right? So if we want to change the reagent, for example, we want to change the T cell purification, magnetic beads, CD4 and CD8. So to what stage we can say they are compatible? So it's after we get the purified T cell and they are compatible, or we should all the way go down to the T cell activation, lentivirus transduction, or the final CAR-T product. So then also for the cell therapy, the sample is limited. And also, usually regulatory agency said you should better to use the patient sample to do the assay development or comparability study. So because hair cell donor, the phenotype is totally different from the patient. So how you use the healthy donors data to prove the change is compatible is also a challenge.
Yeah, so on that note, I would like to hear from Dr. Zhao, right? So exactly, right? So Dr. Jiang, you're very spot on. So when we do potency assay, if you think from a reference standard point of view or the assay, right, point of view, healthy donors. So how much do we take that as the holy grail to demonstrate analytical comparability?
Yes, I think first of all, the changes is which stage, because we're talking about change, is thinking of the impact, right? So I think it's just for the potency assay. Yeah, so normally the regulators recommend to retain the clinical material if it's possible. So, and then that means you can do the side-by-side comparability study. So using the patient one, or you can establish a reference standard. But some of the reference standard can be made for the analytical. It's like a control. Yeah, and then I think even the WHO and also try to get involved in this area. When I was working in the MHRA, I lead the team of 40 laboratories, made the two of the lentiviral vector international standards. So this kind of materials is quite useful. It's very useful. It's harmonize the whole area, right? But if for the potency assay, the main things is you need to know the product and mode of action. Yeah, so that's, yeah.
Yeah, I think that's really, what we read from the guidelines. However, we know as much as we understand the biology. I think there's still a lot of debate about what does stemness really mean, right, in terms of in vitro assay and things like that. So over to you, Dr. Kidd. I want to hear from you. I understood that you have treated more than a dozen patients in Thailand, and there must have been changes along the way, I suppose. Could you share with us what actually works, and hopefully nothing didn't work?
Yeah, okay. So for our case, right, for Genepeutic, although we act as CDMO and also we also have a clinical trial, or we act as a clinical stage with our own CD19 CAR T cell as well. So we actually go a bit different route compared to what has been approved, which is allogenic. Although it's not a universal available CAR T, but we actually collect cells from healthy donor. It's still personalized, so we transduce those cells and then give it back to the patients. Obviously, I mean, there are a lot of concern of like GVHD, graft-versus-host disease, blah, 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 but that can actually be discussed a bit later. But so what I'm trying to say is that, sorry, it just lose my mind. What's the question again?
So comparability, your comparability strategy,
yeah.
What works and what doesn't?
Okay, so with that, during the process of our clinical trial, right, it just happened that because we use, this is the real world sample, so we use human AB serum as a supplement, which is one of the very difficult reagents to actually find. So there's a case where the vendor that we actually originally did the validation studies from discontinued the GMP grade human AB serum. So what we have to do is we have to find an alternate source. So we have to actually do a comparability studies in order to change from one vendor to another, because I mean, obviously changing the vendor meaning changing the source of the human AB serum, right? So that is one of the case, and we did, obviously, in order to do that, we have to change the CMC part. We submitted to the Thai FDA for approval, and then not only that, not only the CMC has to be changed, but the protocol for the comparability studies has to be detailed out and then send it over for approval as well before we execute it, before we can continue with the actual clinical processing. So that's one of the case that we have to go through. But just want to mention that with the GMP regulation, I think one of the important part is to actually have an alternate source. So meaning that the comparability studies is not bad in the sense that, obviously you will need to make sure that you have enough of your reagent or from the same vendor that you did the validation from, right? If not, then, you're screwed, because just like, for example, in COVID time, the single-use bag is in a shortage, and what you do, everything get delayed, right? So that will actually impact on your clinical trial status as well. So I think the comparability studies not only helps to make sure that you always get a full supply of the critical reagent, but also will help you to reduce the cost effectiveness part as well, because you compare directly, head to head, like the reagents that cost you, I don't know, $10,000 compared to like a $5,000, but then both products actually give you the same effect, right? So obviously that will help you with the cost reduction part for the CGT as well.
Yeah, that's a really good point. I think just in CMC language, what you guys did is actually to front load a lot of studies and the scope to expand your design space, right? So in terms of material attributes and sources of that. Now, before we open the floor to pigeonhole questions, Zhao, so you mentioned a few strategies, right? Connecting non-clinical, clinical CMC changes along the way. But if you quickly summarize like 2 practical guidance, right, to the audience, what would be those guidance?
Yeah. So I think if you look at the approvals for 2024, there's already been 2 rejections. We call them CRLs, complete response letter. And the 2 rejections have been CMC related. I mean, the companies came out themselves to talk about it. CMC is going to be very key for cell and gene. And I think comparability is going to be really, really important for us to make sure that we do it well. The first one is, I think what one of my esteemed panelists have mentioned, is retains. Retains, retains, retains. I think that's what my non-clinical head always tells me about. Whether it's your non-clinical, whether it's actually a clinical trial, always keep retains so that when you make manufacturing changes, you can go back and see whether the process is still safe and efficacious. The other thing that's important, if you look at what FDA's guidance saying in terms of comparability, the 2 main things to determine what the scope of the comparability is firstly, how big the change is. Is this a huge change or this is a minor change? And the other one is, what phase are you working on when you make this change? Do not do it in your, okay, I cannot say, you cannot say, try not to do it in your pivotal, your BLA, and your post, because those will require a much larger comparability study to actually make sure that this is going to be accepted by the regulators. Which is why I kind of go back. I was previously in a biotech company. I feel the most important thing to plan for your comparability, to plan what you intend to do for each phase. I've seen folks who say that I've done my IIT in phase 1, I'm going to move to a pivotal immediately for my phase 2. It's not impossible, but it's very challenging. If you've never run the process and you want to move immediately to a pivotal trial, there are changes, and then how are you going to justify these changes to the FDA? I think these are things that you need to consider when you plan for your clinical trial. To me, try to front load a lot of the changes you want. That's what Kitipong has done. Make sure all these things are done before you start your clinical trial and know what you want to do for each phase.
Yeah, thanks, John. So maybe quickly cover these 2 questions. The first one, I believe Dr. Tsutomu has already covered, right? So, is it like point of care, or is it like a centralized manufacturing? Just maybe one other panelist, anything to add on top of what Tsutomu has shared earlier? Dr. Zhao, please.
Yeah, I think maybe we should remember that for the regulator, the point of care, the bedside manufacturer will never replace the central manufacturer process. So that's why if you apply for this point of care, you must demonstrate you definitely cannot make the product in the central facility. So that's the whole thing. It's not say or a central hub. It just depends on. I think a point of care definitely is a future in a way, because one is the product, you don't need to cryopreserve. And then we all know the cryopreserve of cells will damage the cells. Yeah, and then also a lot of the company, I think the ICH done the kind of a questionnaire, asked the people. I think more than half of the people, 50% of the manufacturer, they will do the bedside reconstitute the cryopreserved cells. That is already a point of care. So that is already introduced variation. So yeah, I think it is a future, and will maybe like the sustainable manufacturer, maybe you don't need to transport the cryo product to patient to hospital. So that's maybe like an energy kind of the benefit.
Yeah, thanks, Dr. Zhao. Maybe perhaps cover the last question. It's really asking, you know, what is the new, is there a new partnership to do CDMO? Because CDMO folks, they very rarely, right, get the feedback from the sponsor, right? Their customer in terms of the clinical readout, so they have no ground to fine-tune the process if needed. Perhaps Dr. Jiang, right? I know GenScript, you guys have.
So I will first comment on the first question. So because the technology development, so point of care is possible because they have like in vivo CAR-T, right? So I look at the article, they are trying during the apheresis collection, they do the genetic modification during the process and put the product into the patient. So if the technology is advanced, I think point of care is like possible. So it's through automation, closure, right? So for the CDMO, so usually for me, like for CAR-T company, so they hardly will to like outsourcing their cell therapy product to CDMO, because the reason is like this is final product. So I can outsourcing my lentivirus or plasmid to CDMO, but my final product, I will manufacture by our company or not by CDMO, right? So, and also if like they don't have the facility or they do not want to like establish facility for the final CAR-T product, they outsourcing the CDMO. So I think usually the CAR-T company will work with CDMO to finalize the process. So there is the issue like the CDMO will not see access to the clinical result. I think it's unlikely, because if the CAR-T company do not work with CDMO company to address this clinical issue, so the manufacturing process will not be success, right?
Yeah, so I agree with, I see your point, right? So the silver line is apparently there's, Dr. Jiang sees a close partnership already existing between the sponsor and the CDMOs. John, I recall you have a slide, right, in terms of insourcing, outsourcing. We are not going to elaborate due to the time constraint, but I think that's really needed, right? So I think to your point, the process is the product. This is very true to cell and gene therapies products as well. So I just want to invite all the panelists in one sentence, right? Bring everybody home. What is your wish, or what do you want to achieve in terms of expediting life-changing ATMP therapies for Asian population?
I think that for me, if you actually want to get approvals for your CGT products, I think the one thing is to keep a close watch of your CMC. I think that has been not placed on a high priority for many of the past few years. People just think about the clinical data, but if you want to get approval, focus on CMC, because I think that's going to be one of the gating items for your approval.
I think the key take-home message that I would like to give to the audience is the build the talent pool. It's not only in the manufacturing side, but all the way up to the very early drug development process, because I think since we are venturing into the drug development process, it's very important for the upstream people to understand the downstream. So I think the talent building is quite important. Thank you.
For me, well, I guess treating make it simpler, shorter, and cleaner, I guess, because the production process at the moment is too long, and I have like patients actually die on me while the manufacturing process is still going on. So I think at the moment the technologies has to be actually much simpler than now, and trying to make it shorter so that we have product out from vein time to vein time at a much shorter period compared to now. Sorry.
Okay, I think for the regulator, I think the main key concern is the product safety. Yeah, so I think maybe that's what I would hope can make it a more safe product and then cost less. So to achieve that, maybe need to be a harmonization, the regulation, and the exchange of experience from all agents or regions.
So ATMP is a still new area. So I think process and their facility require the more automated and the new technology should be developed. And then also automated and also data integration must be needed for the future. Yeah, so I think we need to more discuss in Japan COP and the US and the APAC COP. Yeah.
I think the advancing process, make automation, modularize the automation, so use the closure system. So use the low grade at the environment level, C level plus A, so to decrease cost. So it's the key.
Yeah. Let's thank all the panelists for sharing their insights. It's really wonderful. Thank you very much.